Welcome to the Institute of Cellular Biology and Pathology Nicolae Simionescu from Bucharest, Romania. Here in the Viral Transduction Laboratory, we show a part of the adenoviral particle production using an optimized methodology based on the pAdEasy system developed by Professor Vogelstein. The main steps of the technology for producing these adenoviruses are 1. Recombination of pAdTrack containing GFP with the pAdEasy 1 plasmid in BJ5183 bacteria. 2. Packaging the adenoviral particles. 3. Amplification of the adenovirus in AD293 cells. 4. Purification of the adenoviral particles from cell lysate and culture medium. 5. Viral titration and functional testing of the adenovirus. Here, we will present an important step of the methodology, the purification of the adenoviral particles from cell lysate and culture medium. The purification of the adenovirus starts with harvesting the virus-producing cells and the medium. AD293 cells were transfected with the recombinant plasmid, and thus the adenovirus was packaged. Then the adenovirus was amplified by successive larger cultures. Here we obtained 25 T175 plates, and we started the purification of the adenovirus from the cell lysate and the culture medium. First, we checked the green fluorescence of the GFP expressed by the transduced cells. If the cells are still attached, we would detach them by tapping the culture dish or by using a long scraper.